You're listening to Conferences Online Allergy from Children's Mercy Hospital in Kansas City, Missouri. Today is December 4th, 2023. We are doing Abbas Chapter 2, Cells and Tissues of the Immune System, presented by Dr. Nikita Raje. She's an Associate Professor of Internal Medicine and Pediatrics and is the Chief in the Section of Allergy Immunology at Children's Mercy Hospital.
We are talking about Chapter 2 from Abbas textbook, Cells and Tissues of the Immune System. And I'm Nikita Raje from Children's Mercy, Kansas City, University of Missouri, Kansas City. Today is December 4, 2023. We are on to Chapter 2, finished Chapter 1 last week. And let's see if we can start. Okay, so here are my disclosures, nothing pertinent to our talk today. Learning objectives for today are to recognize the structure of various immune cells, function of immune cells, and then the differences in the structure of various lymphoid organs. We'll be talking about various cells of the immune system today and the anatomy and functions. So this is our outline. Couple of pre-test questions. A drug that blocks the function of chemokine CCR7 would result in which of the following? And in 22q deletion syndrome, the thymus fails to develop. Which of the following characterizes the immunodeficiency state in this syndrome? All right, let's get started. We'll come back to those questions at the end, unless you have some other question. Hang on to those answers. All right. So let's start with just the cells, right? The cell counts here are shown, what are the normal ranges. And as we know, neutrophils make up 40 to 60% of those cells, and lymphocytes make up 20 to 40%, and then few of the monocytes, eosinophils, and less than 1% are basophils. We'll talk about the neutrophils, which are also called polymorphonuclear phagocytes, as the picture shows. There are several lobules in the neutrophils, which gives them that name of polymorphonuclear phagocytes. These are cells that are produced in bone marrow from myeloid monocytic precursors, about 10 to the power of 11 every day. So huge number of neutrophils that are produced every day under the influence of the cytokine GCSF. These are 12 to 15 micrometers diameter with lobulated nucleus, and they have two different types of granules. Specific granules, which make up most of the granules, they have these substances or enzymes called lysozyme, collagenase, elastase. These specific granules do not stain with hematoxylin and eosin, and so no basic or acidic dyes in them, and that's how they get the name neutrophils. They also have these azurophilic granules that have affinity to azure dye, and they have these bactericidal or microbial substances that are work against these microbes, defensins and cathelicidins, as well as enzymes like myeloperoxidase. The lifespan of these cells is pretty short, few hours, maybe up to 5 days in the blood. They may last only for a day or two in the tissue once they enter the tissue. Compared to that, mononuclear phagocytes are a little different. They are also produced from the bone marrow and then go into the circulation and end up from being monocytes to being macrophages in the tissues. But also there are these tissue resident macrophages, which are a little different. Instead of coming from the bone marrow, they are produced in the fetal life from yolk sac, fetal liver, and over time they differentiate into these macrophages that stay in specific tissues and get a specific name based on the tissue they live in. Microglial cells, Kupffer cells, dermal macrophages, alveolar macrophages, and so on and so forth. So as you see, this figure basically shows where the macrophages that are found in various locations are produced from. So these monocytes or mononuclear phagocytes are produced under the influence of MCSF, and are 10 to 15 micrometers in diameter. They have this bean-shaped nucleus and have a finely granular cytoplasm. They also contain several substances like lysosomes, phagocytic vacuoles. They have cytoskeletal filaments, and monocytes circulate in the blood. Once they enter the tissue, they become macrophages. Monocytes are have these particular markers on their surface. So CD11b and CD86, MHC class 2, all of these markers are on them. Also, they have some other markers that are used to classify them as classical and non-classical monocytes. Classical monocytes are what we know of as monocytes. They are CD14 highly positive, CD16 negative, and also have CCR2 on them. They are the ones that are recruited from the blood to the site of injury or infection and release inflammatory mediators. Non-classical monocytes, on the other hand, are CD14 dimly positive, CD16 positive, and CX3CR1 positive. They crawl along the endothelial surfaces. This phenomenon is called petroling, and that helps in contributing to tissue repair after injury. So these are non-classical monocytes. Instead of causing inflammation, they help in tissue repair. Another intermediate set is CD14 positive and CD16 positive, is also inflammatory in nature. So there are two types of activation for macrophages: classical activation and alternative activation. Classical activation is what we see as monocytes being activated and getting into the tissue. Macrophages, once they come across the microbe or something, that an antigen that can interact with it and activate it, it becomes an efficient killer. Versus alternative activation, when there are cytokines that activate macrophages that do not cause inflammation. In those cases, they will undergo alternative activation and help in tissue repair. So one of the concern is, can these macrophages identify an antigen which is a self-antigen and cause problem? And typically that doesn't happen because the healthy host cells have a inhibitory receptor on their surface that prevents the macrophages from engulfing our own cells. That's called SIRP alpha, and that recognizes CD47 on our self-host cells, because of which the macrophages will realize that those are not to be engulfed. Function of macrophages based on what kind of stimulation activates the macrophages. They can release cytokines that cause inflammation. They can use their enzymes to kill the microbes. They can also help in tissue repair and fibrosis by alternative activation and help in resolution of the inflammation. So we looked at two different phagocytes, neutrophils and macrophages. This table summarizes the differences between these two. Big ones are short-lived neutrophils, long-lived macrophages in the tissues. They help in basically phagocytosis and then use some of the mechanisms to kill the microbes. Neutrophils do, versus macrophages can also do that, but they also help in presentation of those antigens to T cells. They can act as antigen presenting cells. Neutrophils can undergo what is known as net formation. These lead to extrusion of the nuclear contents of the neutrophils if they cannot engulf the whole organism, if it's a larger organism like some parasites. In those cases, the neutrophilic substances are extruded onto on that microbe so that it can kill it. versus macrophages use their cytokines for some of the actions that they perform. Also, macrophages lead to caspase-1 activation that can lead to fever. Moving on to another cell, mast cell, that's also produced from bone marrow under the effect of stem cell factor or c-KIT ligand, which is needed for the production of the mast cells and development of these cells. So again, so anything in red that you see, that's an important memorization point, name, substance. Here, remember, mast cells are produced under the effect of stem cell factor, also has another name for a lot of terms that are used in immunology. As the second years would know, there are more than one name. And here, stem cell factor also has a name, c-KIT ligand. And I would recommend that you try to memorize both those names. All right, so mast cells, we know them pretty well. These are cells that have granules that are filled up with contents like histamine and tryptase, and there are receptors for IgE on their surface. So when the IgE antibody binds to those receptors on the surface, these granules can release these substances, histamine and tryptase. These are typically present in the tissues. So if there are not many in the blood circulation, so you don't typically see them on the blood counts because there are very few of them in the circulation. These are typically found in the tissues and close to the neurovascular bundle. Basophils, again, produced from bone marrow, very few of them, but they are in the blood compared to mast cells, which are more in the tissues. They also contain granules that bind basic dyes and have similar mediators like mast cells, again, express IgE receptors on their surface, and pretty much the function is very similar. Basophils more commonly found in blood, mast cells more commonly found in the tissues. Eosinophils, again, produced under the influence of GM-CSF, IL-3, IL-5. These contain granules. Unlike basophils, they contain basic proteins, which means they bind to the acidic dyes, and they are present in the tissues as well as blood. They are present in the tissues in the mucosal linings as well. Dendritic cells, there are two different types, or actually three different types. Classical dendritic cells, plasmacytoid dendritic cells, and then of course the monocytes can also come from dendritic cell precursor, monocyte precursor. So these are produced from the bone marrow, but there are some that are produced from the fetal liver, and they go on to become specialized dendritic cells like Langerhans cells in the skin. One of the features that's important to remember about Langerhans cells is they have something called Burbeck granules. Okay. So like the other cells, dendritic cells need to be produced under the influence of a cytokine. So here, this is FLT3 ligand that helps to produce more dendritic cells. These are cells that have long membranous projections, and that's how they get their name, dendritic cells. The typical dendritic cells are called classical or conventional dendritic cells. They are present in the tissues, and then they are pretty much waiting to use those projections to sample what's around them. And if they come across something substantial, they will process them. They travel to the lymph nodes to present those antigens to the T cells, so they are antigen presenting cells. There are also another type of dendritic cells. Important to remember these, important to remember their function. They are called plasmacytoid dendritic cells. They are early responders to viral infection, and they produce interferons, type 1 interferons, and they are almost factories of interferons. So that's all they do. They produce a lot of type 1 interferons that helps in responding to the viral infections. Some pictures of dendritic cells. Um, and basically, the table here gives you the differences between different types of dendritic cells. Again, something to remember, there are various there are various types of dendritic cells that have various surface markers. So CD11B, CD11C are important to remember. Langerhans cells will have the langerine, CD207. They'll have the Burbeck granules. The plasmacytoid dendritic cells have high levels of toll-like receptors on their surface, and that's how they recognize these are toll-like receptors for viral um, uh, PAMPs, and because of that, they are able to recognize the viral infection and produce a lot of type 1 interferons, compared to other dendritic cells, which are gonna be used to present antigens to the T cells. What are the other antigen presenting cells? Macrophages, as well as activated B cells that help in presenting antigens to T cells. All three of them, dendritic cells, macrophages, and activated B cells, are called professional antigen presenting cells. However, the nucleated cells can also present antigens. So all nucleated cells can use class 1 MHC on their surface to present antigens to CD8 cells. So they also are antigen presenting cells. But when we talk about professional APCs, we are talking about dendritic cells, macrophages, and activated B cells. Similarly, follicular dendritic cells that are found in lymphoid follicles are antigen presenting cells, so they present antigens to B cells. Moving on to lymphocytes, as we know, they are produced in bone marrow. 65% of the lymphocytes found in the body are actually in the lymphoid organs. So even though they make up about 40% of the blood cells, only 2% of lymphocytes in the body are found in the blood. So others, lymphocytes, majority of them are in lymphoid organs, mucosal associated lymphoid tissue, bone marrow, skin, and then 2% of those are in blood. These are clonally distributed, they have diverse and specific antigen receptors, and as we know, they are either B cells or T cells, and we'll talk about the innate lymphoid cells as well. Talking about B and T cells, B cells, the ones that we typically talk about, are called follicular B cells. There are also other two types of B cells called marginal B cells and B1 B cells. T cells, the ones that we majority of the times that we call T cells, are the alpha beta T cells. And these are the ones that we think about as CD4 positive, T helper cells, CD8 positive, cytotoxic cells, or Tregs. Most of these are CD4 positive, but there are CD8 positive Tregs as well. 5% of our T cells can be gamma delta. And then there are some other subtypes of T cells like NK T cells. So tables showing more features about these cells. Again, not going too much into detail. I just wanted to point out the majority of these alpha beta T cells are going to be CD4 positive or CD8 positive, so single positive cells. And again, Tregs that are FOXP3 positive, apart from being CD4 positive, they also have CD25 on them. NK T cells, on the other hand, have the markers for NK cells as well as T cells. So again, they have alpha beta TCR on them. CD56 and CD16 are the markers for NK cells, along with the T cell marker CD3. So they have all three on them. Gamma delta T cells can be CD3 positive, but can be variable for CD4 or CD8. So they can be double negative. They may not have CD4 or CD8 on them. And then there are cells called MAIT, mucosal associated invariant T cells. And these are also alpha beta positive. Most of them are CD8 positive. But B cells, the majority of them are follicular B cells. And these are the ones that typically produce all types of diverse immunoglobulin classes compared to marginal zone B cells, which have very specific, very limited specificity of their BCRs and produce mostly IgM. They are and then there are B1 B cells, which are also mostly producing IgM and have limited specificity compared to follicular B cells, which have diverse specificities. So we know that regardless of whether they are going to be B or T cells, they come from the bone marrow. The ones that are destined to become T cells go to the thymus. The B cells stay in the bone marrow and mature up to they develop into immature B lymphocytes and then are released into the periphery where they complete their maturation, versus the T cells complete their maturation in the thymus, and then the cells that are released into circulation are mature naive T cells. So various stages of the B and T cells, they start out as mature naive cells. When they come across the antigen that they can interact with, they become activated lymphocytes. Once they are activated, they proliferate and differentiate into effector lymphocyte. So for B cells, that would be plasma cells. For T cells, that could be subsets of various types of T cells. And then these plasma cells can produce antibodies. The helper cytotoxic T cells can give that short-term effect from their effector molecules, but they can also become memory lymphocytes, either of them. So all four stages are of the lymphocytes are important to look at. And something to remember is how would you recognize them? So they have various markers at different stages of their lifespan. Important to remember for naive T cells is they are highly positive for L-selectin or CD62L, CD45RA, and CCR7, right? So these are the three markers important for naive T cells. That's how we recognize naive T cells in lab. So the cells that are positive for CCR7, CD45RA, and L-selectin are called naive T cells. Cells that have CD45RO on their surface, they lack L-selectin and they lack or are low on CCR7, are the typical activated or effector T cells or memory T cells. As you look at some of these, you will realize that some of the activated and effector T cells, the effector T cells and activated T cells will have CD25 on them. They will have the CD45RO on them, whereas memory will be low on CD25 and will still have CD45RO on them. So some of these markers on the surface will help you determine what cell you are looking at. Similarly, B cells have various markers on their surface. So CXCR5 is positive on naive B cells, but they are low on CD27, whereas activated or memory B cells will have CD27 on them, and that's a marker for your either of those classes. We typically use it to define memory B cells along with some other markers. So they also have on their surface IgM and IgD if they are mature. So mature naive B cells will have IgM and IgD on their surface. Compared to activated effector or memory B cells, they may have switched their class, and so they may have G, A, or E on their surface. So just kind of going into some more details about that, naive lymphocytes are mature B or T cells. So they have completed their maturation process. However, they are inexperienced, which means they have not come across the antigen that will activate them, but they are fully mature. They die after 1 to 3 months if they have not come across the antigen of their choice. Naive lymphocytes are smaller in size. They are in G0 stage of cell cycle. And once activated, they'll get into that cell cycle and they will grow in size. They do need some stimulation, even when they are not seeing the antigen of their choice. So they get weak recognition from self-antigens and some cytokines. So for T cells, that comes from IL-7, and for B cells, it comes from BAF. Important to remember, BAF, B cell activating factor, or BAF, is an important survival factor for B cells. Anyone know what the other B cell survival factor is called?
Sure,
Okay.
Dr. Raje.
Okay, keep thinking. All right, and then we talked about how CD45 CD45RA is present on naive lymphocytes. It's a marker of naive lymphocytes. This is an isoform of CD45, which means that the gene for CD45 can make two different isoforms. One of them contains exon A, and that's called restricted A, so which means it's present, that exon A is present in that isoform. So that isoform is called CD45RA. CD45RA recognizes naive lymphocytes compared to the memory lymphocytes. We'll look at it in a minute. Before that, we can talk about the effector lymphocytes. These are ones that get activated, proliferate, and then differentiate into effector cells. And they express different surface molecules. For helper T cells, CD40 ligand is expressed. Anyone know what the CD marker for that is? So again, important to memorize the CD
marker for which one?
CD40, sorry?
CD marker for which one?
CD40 ligand.
CD154.
So CD154 is correct. You have to memorize both of these names. CD40 ligand is the most common name that we hear all the time. You have to memorize the CD marker for it, CD154. And then CTLs express cytoplasmic granules in them, so granzyme and perforin and things like that. The effector lymphocytes also express CD25, which is the activation marker for T cells. These are short-lived and not self-renewing. Compared to, for B cells, the effector lymphocyte is called a plasma cell. It has a very specific structure. It has an eccentric nucleus that gives away the pattern. It has chromatin around the nuclear membrane in cartwheel pattern. And of course, it secretes thousands of antibodies every second. Plasma cells develop in the lymphoid organs, and then they migrate to bone marrow and live there and secrete the antibodies. Plasmablasts are precursors of long-lived tissue plasma cells. They are found in blood in really low numbers. So here's the eccentric nucleus and the cartwheel pattern that you see in the plasma cells. Okay, memory lymphocytes, these are slow cyclings. They are present in slow cycling state for months to years. And they again have specific markers on them. So if you think about memory T cells, they have CD45RO. What does the RO stand for? Exon A is spliced out, and that RO, the O is for out. Basically, the restricted A exon is spliced out in these T cells. They also have IL-7 receptor on them. IL-7 also has a name CD127, right? So same way, the naive T cells also have IL-7 receptor on them. So there are some, so IL-7 positive CD45RO are memory T cells. IL-7 positive CD45RA are naive T cells. Memory B cells have CD27 on them. They also have membrane IgG-AE on their surface. So as a child grows, the number of naive T cells that they are born with go down in number, and percentage, whereas the memory T cells go up in number. All right, so we talked about T and B cells. Another type of lymphoid cell is called innate lymphoid cell, or ILCs. ILCs are derived from bone marrow. They are similar to lymphocytes. Their effector function may be similar to T cells, but they lack the TCR. They do not have TCR on their surface. They are actually important for innate immune response and recognize and eliminate stressed and damaged cells. So different types of ILCs, ILC type 1, type 2, type 3. These are the ILC type 1 are natural killer cells, but there are other types of ILCs. Depending on their class, they may secrete various other cytokines. All right, so that was a discussion about different types of cells. I'm going to stop here and ask for any questions. All right, anyone thought of what the B cell survival factor, another one, is? I'm going to just grab my water. I'll be back.
I'm not sure if this is right, but I remember like BAF in April. Is it April?
Want to get the water?
Yeah, April is correct. So BAF and April are two B cell survival factors. BAF is important for, I think it just depends on what stage of B cells we are talking about. Any other questions? Okay. Let's talk about lymphoid tissues. And I think I went through the cells pretty quickly because most of the times what happens is that discussion goes on for long and then the lymphoid tissue discussion has to cut short. And I really wanted to pay more attention to lots of important concepts in these for the anatomy and function of the lymphoid tissues. Lots of board questions that can come from this as well. So pay attention. So we know that there are two types of lymphoid tissues, primary and secondary. Primary or central lymphoid tissues or generative organs are bone marrow and thymus. And then peripheral lymphoid tissues or secondary lymphoid tissues include lots of different ones: lymph nodes, mucosal immune system, spleen, cutaneous immune system, and then there are aggregates of lymphocytes and various organs. So let's talk about the bone marrow. We know all the blood cells are generated here. At birth, all the bones will have bone marrow, but by puberty, bone marrow is present only in the flat bones. So that's sternum, vertebrae, iliac bones, ribs. So there's part of the bone marrow that's called red marrow that is sponge-like reticular framework between the long bony trabeculae. And the spaces in this framework is the one that contains the network of blood-filled sinusoids. So there is some endothelium, but instead of like a blood vessel where the endothelium is continuous, there is discontinuous basement membrane. Outside the bone marrow, there are clusters of precursor of B cells in various stages of development, and then there are mature fat cells. So as the blood cells are produced, they mature, and then they migrate through the sinusoidal bone marrow and between endothelial cells in the vascular circulation. We all know this chart pretty well. This is the chart for hematopoiesis. We know that the stem cells self-renew and then produce multipotent progenitor that turns into progenitor, which can be myeloid or lymphoid. And then depending on the type of cell produced, they can differentiate into cells that are going to give rise to T cells, B cells, and K cells for lymphoid cells, and again, or ILCs from lymphoid progenitors. So they become pro-B, T, and K cell, then become pre-T or B cell for B and T cells, and then produce T or B cells. Same way for the committed precursors on myeloid side, various different types. I'm not going to go through each of these names. I think I want to make sure that you understand this lower part of the picture here, that initially the stem cells are the ones that are that need the stem cell factor. Remember, the second name for stem cell factor is c-KIT ligand. And then those markers go down, and then there are lineage specific markers that are more prominent as the hematopoiesis progresses and the cells differentiate into various cells. So the lineage independent growth factors are important for the initial precursor stage, but later it's the lineage specific growth factors that become really important. And then so the cell division becomes more prominent again when these precursors proliferate. Another table that tells you what cytokine is important for maturation of what kind of cell. So stem cell factor, c-KIT ligand, important for stem cells. IL-7, important for T cells. GM-CSF, important for granulocytes and monocytes. MCSF for monocytes. GCSF for granulocytes. IL-3, important for all the cells. IL-7, I already talked about. FLT3, important for dendritic cells, also for B cells. Let's talk about thymus. Thymus is a bilobed organ. It's present in the anterior mediastinum, and each of the lobe has multiple lobules. And that lobule is divided by fibrous septa that you see here, right? And each lobule has an outer cortex and inner medulla. Cortex is dense with lots of lymphocytes, mainly T cell collections. Medulla has sparse lymphocytes, but also has macrophages, dendritic cells. It also has non-lymphoid cells. The non-lymphoid cells include epithelial cells, and these could be cortical epithelial cells, cortical thymic epithelial cells, or medullary thymic epithelial cells. And then there are structures in the thymus called Hassel's corpuscles. These are rows of epithelial cells in the medulla and lots of degenerating cells that make up these corpuscles. So again, medulla and cortex. Cortex is the dense region, medulla is the sparse region. Lots of T cells, developing T cells in both the areas, but the medulla also contains several other types of cells. Cortex also has different other cells, but just we'll talk about how they function and so why it's important to know all those cells. So lymphocytes come from the bone marrow that are destined, if they are destined to become T lymphocytes. They are in various stages of maturations. When they enter the thymus, they are called thymocytes. At that time, they do not have CD3 on them. They do not have CD4 on them. They do not have CD8 on them. These are immature thymocytes that come into the cortex, and as they go through the cortex, enter the medulla, and by the time they come out of the medulla, they become mature. So in the medulla, there are still thymocytes that undergo various stages of maturation, and at the end of that maturation, the naive mature T cells exit the thymus and enter the blood circulation. Lymphatic system is made up of lots of lymphatic capillaries, start out as capillaries that have overlapping endothelial cells with one-way valve, but they do not have a tight intracellular junction or basement membrane, so they permit free uptake of interstitial fluid from all parts of the body. That lymph flows through the lymphatics due to contraction of the smooth muscle cells, and these lymphatics are arranged in series, right, to lymph nodes. So they move on and go through the lymph nodes and then converge into larger lymphatic vessels, finally end up into thoracic duct or right lymphatic duct and into the superior vena cava, where 2 liters of lymphatic fluid is dumped every day. So along these lymphatics are the lymph nodes, and they are an important secondary lymphoid structure. So lymph nodes are lymphoid structure that has a capsule, unlike the thymus, which we don't know too much about how it functions, some of the structures of the lymph node are pretty well defined. It's an encapsulated vascularized lymphoid organ. And as you can see, there are these efferent lymphatic vessels that bring in the lymphatic fluid. This lymphatic fluid is draining various parts of the body. So if the antigen entered that part of the body, the antigen might be brought in by the APC or by other means like complement and things like that from the blood. So blood in different places or the efferent lymphatic vessel using the antigen presenting cells. So that's where the antigen enters the lymph node. This efferent lymphatic will bring in the lymphatic fluid that goes into the subcapsular sinus, and then it goes through the medullary thymus, sinus. And from the medullary sinus, as it goes from the subcapsular sinus, then the medullary sinus, then it goes into the efferent lymphatics. So there are two parts of the two zones of the lymph nodes called B cell zone and the T cell zone. The B cell zone typically will form a structure called follicles, right? The follicles can be primary or secondary. What does that mean? In the primary follicle, there are B cells that are in various stages of maturation in the for the B cells, but the B cells have not come across an antigen that's specific for those B cells. When the B cell gets activated, it forms a germinal center in the follicle, and it's then called a secondary lymphoid follicle. So the germinal center has a dark zone where there are proliferating B cells. The cells that got activated will proliferate a lot and form the centroblasts, versus the light zone is where there are B cells that have or centrocytes that have stopped proliferating. So it has a dark zone and a light zone in the germinal center. When you see a germinal center, that is called a secondary B cell follicle, versus they start out as primary follicles. Compared to the follicular structure, the T cells are actually next to it. So they are in the paracortex. So in the cortex are these follicles, and the paracortex is where the T cell zone lies. Hopefully you can see my cursor here. We are talking about the T cell zone. That's the paracortex. It's organized in cords. So let's talk about these structures again. So B cell zone is formed around follicular dendritic cells. These follicular dendritic cells have nothing to do with the dendritic cells that we just talked about. So dendritic cells, which are the APCs or plasmacytoid cells, are dendritic cells that are coming from the bone marrow, right? Compared to those, the follicular dendritic cells are completely different. They have nothing to do with these dendritic cells, but these are dendritic cells that are like trees. They are plucking out basically the antigens that are coming to them from, say, the blood, bound to tagged by complement receptors or antibody receptors. And so they pluck out those antigens and express them on their surface so that the naive B cells can look at those. They are presenting those antigens to the B cells. In the primary follicle are naive B cells. When the B cell finds the antigen it's specific for, those B cells proliferate and form secondary follicle. So a germinal center is created, and that's where they give rise to high affinity antibodies. They produce plasma cells or memory B cells, and those can get out of the circuit from the lymph node and go back to the bone marrow, where the plasma cells can reside and produce antibodies. Now let's talk about the paracortex. Paracortex is formed around fibroblastic reticular cells. So FDCs are in the follicle. Those are the ones where the B cells reside, and FRCs are around the T cell zone where the T cells reside. So beneath and central to the follicles are these networks of FRCs that form FRC conduits. They form a tube-like structure. And these FRC conduits contain extracellular matrix like collagen fibers, mesh of fibrillin microfibers, and they are surrounded by a basement membrane that's produced by FRCs. So that forms like a tubular structure. And these conduits extend from subcapsular sinus to the medullary sinus. So these FRCs that give rise to this tubular structure is where the T cells are going to come across, because FRCs are the ones, let's see, are the ones that give rise to these chemokines. I guess that's what it comes to. Okay, chemokines that help to attract T cells to that zone. So what's that chemokine? It's called CCL19 and CCL21. So CCL19 and 21 that are produced by these FRCs in T cell zones will attract cells that have CCR7 on them. To begin with, the naive T cells are the ones that have CCR7 on them when they are released from the thymus. So from the thymus, when they come out, they are going to attract it to a place that they find CCL19 and CCL21 on. Dendritic cells that enter the lymph node through efferent lymphatics also express CCR7. So dendritic cells also go to the T cell zones. So the dendritic cells acting as APCs and naive T cells get a time to interact in that T cell zone where they can express the antigen. Naive T cells can come across that antigen. And if it's a specific antigen for that naive T cell will get activated. Versus the B cells, naive B cells that are released from the bone marrow, they enter the lymph node through the HEVs, right? So these lymphocytes are entering the lymph node through high endothelial venules. And the naive B cells express CXCR5 on them, so they are attracted to a place that releases CXCL13. This CXCL13 is produced by FDCs in the follicle, so naive B cells get attracted and know where to go when they enter the lymph node. They go to the B cell zone. So again, FDCs in the follicle are important, release CXCL13, attract cells that have CXCR5 on them. FRCs in the T cell zone, they express CCL19 and CCL21 and attract cells that have CCR7 on them. All these names in red are really important to memorize, really important concepts to memorize. Also important to keep in mind is these receptors and chemokines maybe they are more important for specific locations, but the cells have receptors, and whenever they have that particular receptor, they can go to that particular anatomic place. So CCR7 is not exclusive to naive T cells. Any cell that will express CCR7 on them will go to the T cell zone of the lymph nodes. All right, so we talked about how the lymphocytes end up where they end up. Now let's think about how the antigen comes where it's supposed to come in the lymph nodes. So antigen is actually sorted by molecular size. So in the floor of the subcapsular sinus, there are certain macrophages, sinus macrophages, that take up really heavy, so high molecular weight antigens. So microbes and antigens they might be just taken up as they enter the subcapsular sinus by the sinus macrophages. But the low molecular weight antigens, they can get out of the sinus through the FRC conduits. And they are passed on to resident cortical dendritic cells those cortical dendritic cells can take up those antigens and present those antigens. All right, that's all for the lymph nodes. Now let's talk about spleen. So you got the basic idea of how every lymphoid organ has T cell zone and B cell zone. And it has those follicular structure, which is specific to B cells, and the T cell zone is pretty much next to it, but it's not in a follicle. The structure is not in the follicles, right? So for spleen, there are some differences. Let's look at the spleen as a whole. It's a very vascularized organ, right? It has red pulp and has white pulp. Red pulp is blood-filled vascular sinusoids. So all the blood pretty much can go in there. It acts like a filter for the blood. Whereas the white pulp is where the lymphocytes are going to come in. So the blood enters through the splenic artery and divides into branches and then divides further into vascular sinusoids. The sinusoids are lined by macrophages that can take up the big antigens. And then eventually the sinusoids are going to end up into venules that carry blood into the splenic vein, right? This is red pulp. We are talking about red pulp, which is not what we are going to concentrate on. Our interest is in lymphocytes, where they are going. So lymphocytes are going to end up in the white pulp. The white pulp is surrounding central arteries. So a trabecular artery is going to divide into central arterioles, and that's around those central arterioles eventually actually drain into the marginal sinus. So along those areas is where the white pulp is organized. So the T cells are present in what we call periarterial lymphoid sheath, or PALS. So along the central arteriole is where the lymphocytes that are T cells are going to attracted to, or PALS is where they are attracted to, versus the B cells are attracted to the follicular regions. Also, they are attracted to the marginal zones. So the B cells in the marginal sinus can become marginal B cells, or inside the follicles are the ones that are follicular B cells. And these two types of B cells can sometimes be immature when they come out of the bone marrow and complete their maturation in the spleen. So B cells can end up in the follicles or in the marginal zones, whereas the T cells end up in periarterial lymphoid sheath, or PALS. Really important to remember these facts. So one important thing to remember is there are no HEVs in spleen because blood is freely flowing into spleen. It is a filter for the blood compared to lymph nodes where the blood is entering through HEVs. The lymphocytes are coming from HEVs. Also, there are no incoming lymphatics here because the antigen is delivered by the blood itself. All right, the next, this is basically the last slide that talks about how there are regional immune systems in skin, GI mucosa, bronchial mucosa, and then there are some non-encapsulated lymphoid structures or aggregates of lymphocytes that you find in various different organs. So questions about these features of the lymphoid organs. All right. Let's talk about the questions that we talked about at the start of the chapter. A drug that blocks the function of the chemokine receptor CCR7 would result in which of the following abnormalities?
I think maybe option D, absence of follicular dendritic cells and lymph node follicles. Does it stop dendritic cell migration?
So these are follicular dendritic cells. Remember,
so
there are
they're
two
already
types of
there.
dendritic cells, right?
Right.
So you are right, the dendritic cells will also not be able to come to lymph nodes, but those are conventional dendritic cells that are present APCs for T cells. When you hear follicle word, think about B cells. Follicle anywhere in lymphoid tissue is meant for B cells. So follicular dendritic cells are the ones for the B cells. And so thinking about keeping that in mind, think about CCR7.
CCR7 is related to the naive T cells going into the the movement of cells in the lymph node. So I was going to say reducing the number of T cells in the lymph nodes. So A is what I would have said.
Okay, so that's the correct answer. B cell follicle, the next one talks about B cell follicles or thin thymic cortex, and really CCR7 is mainly a chemokine receptor that's just kind of helping cells to go in a certain location. In this case, it's CCR7, so attracting cells that have CCR7 on them to a region where there is CCL19, CCL21. So I think of these chemokine receptors, and we'll talk more in the next chapter, as GPS. It's just telling you where to go from where. Where are you going to, which place you are going to. All right, second question, 22q deletion or DiGeorge syndrome, thymus fails to develop. What's the characteristic in this immunodeficiency state?
It should be C.
C is the correct answer. Deficiency in T lymphocytes. Thymus is not well developed, so the maturation of T cells suffers, and there is a cell-mediated immune issue.